Good morning and welcome to USI's 14th Endeavor Symposium. My name is Anna Strulja and that is for the Department of Sociology, Anthropology and Criminal Justice. Today I will serve as the chair of session 2 of the symposium. We will start today with Jonathon Fulkerson. He's from the Department of Biology and his paper is titled P53 inhibits 2-chloroethyl sulfide mediated centrosome duplication in SARS-2 cells. It's all Greek to me, but I'm looking forward to understand what this is all about. So, join me in.
Thank you very much. Like she said, my name is Jonathon Fulkerson. I'm a sophomore here at USI. I first got the opportunity to start researching last summer via the EURP, Early Undergraduate Research Program. We worked through the summer and then continued the research on into the fall, wrapped up a particular project, hoping that'll be submitted to a journal soon, and I'm real excited about that. So what exactly have we been working on? P53 protein reduces centrosome amplification in human cells exposed to subtoxic levels of 2-chloroethyl sulfide, or 2-CES. There's three main components to this project. There's the P53 protein, 2-CES chemical, and of course centrosomes. So 2-CES. This is a chemical analog to the chemical weapon mustard gas. As you can see, their structures are almost identical. 2-CES is a widely accepted surrogate. It's a little easier to get a hold of and also safer to work with. Mustard gas is a vesicant, a blistering agent. It's also an alkylating agent, which means that it can damage DNA. Also, extreme significance is that it's a carcinogen. It's known to cause all types of cancers in the upper respiratory tract. Mustard gas has been used since World War I. It's still an issue in developing worlds because it's fairly cheap to synthesize and also not too terribly difficult to synthesize. Not only are those affected via attack, but also those involved in the manufacture of mustard gas are prone to increase the incidence of the cancer. So there's a few different ways that mustard gas may be inducing cancer. Could be through reactive oxygen species or free radicals, could be directly damaging DNA, or it could be involved with alterations in centrosome number or any of the above. We're looking at the relationship with centrosomes. Dr. Bennett's lab previously showed that mustard gas does indeed induce centrosome amplification, which I'll get more to that here in just a moment. So let's take a moment to talk about centrosomes in particular. The centrosomes are cellular organelle comprised of two centrioles. They're involved in cell shape, cell movement, intracellular support, and what we're focusing on is organization of the bipolar mitotic spindle and how it directs the equal segregation of chromosomes during mitosis. In a normal life cycle, during cell division, you'll notice that the chromosomes line up in the center of the cell and two centrosomes organize the equal segregation of the chromosomes so that the ultimate daughter cells have an equal distribution to some of them. Now, if you've got amplification of centrosomes result in more than two, this can result in an unequal distribution of chromosomes, which is called chromosome instability. This can become a pretty bad problem. The reason that this is a serious issue is normally over a cell's lifetime, it would take between 5 and 6 mutations before a cell becomes cancerous. But when you have entire sections of the genome via chromosomes being changed all at once, you're getting an equivalent amount of misinformation in a much shorter time frame. So our body does have some things in place in our metabolism that can help protect against some of these things. The most famous on the cancer side of things is the P53 protein, aptly also called the tumor suppressor protein. Whenever a cell undergoes stress or DNA damage,